plastic particles, mostly focused on the risks to aquatic life. And then we also have a network of now about 190 different organizations spanning academia to the nonprofit sector, industry, as well as government agencies. Just a quick note, we will be recording this meeting today, so let me know if you're not comfortable with that. But since we are in the middle of the summer and a lot of people are on vacation right now, thank you for joining us regardless of it being the middle of the summer. By the way, we're going to make sure we post this on our website so people can access it if they need to. Also, please mute yourself if you're not currently muted, just so we avoid having too much in the way of background noise. So there should be a little mute button in the corner of your screen, or it might be along the bottom depending on your view. So our team leaders here at Oregon State University are Dr. Stacy Harper, myself, Dr. Chris Langdon, and then we have Dr. Wayne Landis at Western Washington University, a little bit up the way in Washington State here from us from Oregon. So we're super excited today to be welcoming Dr. Shelly Moore. She is going to be talking about the California Trash Monitoring Methods Playbook. And Shelly has been an environmental scientist conducting research on aquatic systems in Southern California for over 25 years. She's worked as both a project and data manager for large-scale regional surveys, has developed various applications for scenario and data-driven analysis. And she currently works at the San Francisco Estuary Institute and is as an executive director for a brand new nonprofit, the Moore Institute for Plastic Pollution Research. And so currently her focus, like a lot of us, is on microplastics and plastic pollution and developing monitoring methods. So she's going to tell us a lot about that. And then Wynn Cowger, soon to be Dr. Wynn Cowger, is a PhD candidate at the University of California Riverside. He studies the sources and transport of plastic pollution in the environment, and I believe will be a postdoc at the Moore Institute soon as well. So super exciting. It's all connected. So we'll give them each about 15-20 minutes to talk about their work. And then we'll have an opportunity for a breakout group with each presenter. And so you're welcome to join either one. We'll make it flexible so you can bounce back and forth between the two groups. You should be able to assign yourself to your own breakout room. If you have trouble with that, just let me know, and either me or Stacy can assign you to a room. And then we'll have a short break, and we'll finish up with Emma Sharp, who is a master's student at Western Washington University. She is in Wayne Landis's lab, and she is using Bayesian modeling approaches to assess the risk of, at this point, tire wear particles to aquatic ecosystems. So we at OSU generated a bunch of data in the lab, sent that up to her, and then she's using existing data from other researchers and other sources as well to put together this model. And so we'll get to hear about that. And then last but not least, after the teleconference, and just found out about this a few days ago, there's going to be a hearing on Capitol Hill that you can attend virtually. And Dr. Jen McIntyre from Washington State, who was one of the authors of a recent science paper that looked at the toxicity of the chemical 6-ppd-quinone, I believe it was, in tire wear leachate to salmonids, and found in some cases lethal toxicity from that chemical. And so she's going to be on Capitol Hill today talking to the Natural Resources Committee in the House, if you want to tune into that later on. But it looks like we're getting close to 9:40, so without further ado, I'll go ahead and get on with a few more slides from our end, and then we'll start with presentations. Before we start with our presentations for today, just wanted to remind people of a few interest groups that the Pacific Northwest Consortium has available for people to join to talk about more specific topics related to micro and nanoplastics research. So we have the effects interest group, which focuses on effects, responses of aquatic and terrestrial organisms as well as humans to plastic exposure. And also we really try to link this to occurrence and fate. And like the other interest groups I'll mention, this is a 1-hour journal club that usually meets monthly, skipping this month. And members of that group can volunteer to present papers on various topics of interest, so we try to keep it open. An example is that one of our group members suggested we talk about co-exposure and combined effects from perfluorinated chemicals and microplastics at our next meeting. And so you have some example papers of what that will look like. And so topic for August is plastics and PFAS, and I'm the leader for that group if you want more information. We also have a microbial impacts interest group. And this group is led by Sebastian Singleton and Jared Stein, graduate students at OSU. And they focus on interactions between microbes and microplastics, and also do a 1-hour journal club every month with guest speakers, kind of a similar format. And all of these meetings are recorded so people can view them afterwards if you're not able to join. We have an analytical tools and characterization interest group, and these meetings are monthly on the second Friday, so they'll be having their next one in August. And this group aims to create opportunities for people both inside and outside the analytical realm to really learn about emerging methods and techniques. You know, it's a way to form collaborations. It's a way to broaden your network for, joining any of these interest groups. And this group is led by Manuel Montano at Western Washington University. And then last but not least, we have our science communication group. And this one consists of both internal and external groups. So we have an internal group that's led by John Dickens, who is a grad student in my lab here at OSU. So doing outreach via podcast and social media. Some example pictures we've posted here are from a campaign we're currently running for Plastic Free July. If you're interested in learning more about it, please follow us on either Instagram or Twitter. We have a contest that's associated with the one of the local co-ops here in Corvallis that offers a lot of plastic-free products. And externally, Ellie Jones, who is the leader of our external science communication group, along with Bonnie Monteleone, they're bringing in outside experts to advise on improving science communication and linking us to other groups with events like Scientists and CEOs, which Ellie Jones has organized, with companies that are producing products that have a low environmental impact. So lots of guest speakers, even journalists who give us really great advice on how to interact with the general public and how to better communicate our science to the media, as well as thinking about things like how to design infographics. And so if you're interested in joining any of these groups, you can go to this link here on our website, so interest group link, or you can email specific group leaders to get information or to sign up for those monthly meetings. And so with that, let's go ahead and move on to today's talks. And so I believe first up we have Shelly Moore from SFEI and the Moore Institute. So I'll go ahead and stop sharing my screen and let you share yours.
Sounds great.
All right.
I'm getting host disabled participant screen sharing.
Or hold on, let me fix that.
Okay.
All right, you should be able to now. Sorry about that.
All right. Can you see my screen?
I see. Yes, I can see your screen. Perfect.
Awesome. All right. So I'm going to go really quickly. I've got a ton of information to share. If you want detail on anything else, please ask me in the breakout room. All right. So we're going to go over a number of things. I'll talk about the purpose of the playbook, describe the methods, give you a little taste of what the method evaluations look like, and then describe the method playbook. So let's talk about some of the background. We've had a lot of things happening here in California when it comes to trash and management of the trash, making sure it doesn't get out into the environment. Of course, a lot of places have bans. We've also had some TMDLs passed. The most heard about one is probably the one with LA River. And we also have these statewide trash amendments. And basically there's two different tracks that municipalities can take. One is they can do track one, where they have to install full capture devices, meaning that anything larger than 5 millimeters must be captured before it gets to receiving waters. Or they can choose track two, which is not necessarily full capture devices, maybe partial capture devices in education, more street sweeping, all kinds of other things, but it requires monitoring. So there's a need to have some monitoring methods for those that are choosing track two. In the past, we've had a lot of people doing trash assessments. The same thing has happened with microplastics, and everybody's going off doing their own thing. Oftentimes it's just a shot in time where you go and you take a one-off study here or there, and there's no way to compare the results. So we really need some way of comparing results if we're going to look at regional assessments and whether or not the problem is getting better or worse. Back in April of 2017, there was a stakeholder conceptual meeting in Oakland, California, and there was a lot of talk by different types of stakeholders. We had nonprofits, we had what I call the professional trash people, where they get paid to go out and assess trash or collect trash. We had all kinds of different people, and we wanted to get an idea of basically what the needs were. This was a meeting put on by the Ocean Protection Council and NOAA. And what came out of that meeting was, we want to know specifically about monitoring methods, and we're most interested in riverine areas and rivers and streams. And we want a comparison of some different types of methods that have already happened, that are already being used, as well as we want to look at a new method. So again, the reason for all of this was we want to be able to do these regional comparisons, so spatially and both temporally, and basically ask some different types of questions that tell us whether or not the policies that are being put in place are actually working. So this was kind of a starting point for developing monitoring questions. It's really important that you know what your question is going into your monitoring effort, so that you can make your monitoring method, make sure your monitoring method answers your specific question. So this was a project that happened over 3 years. It was funded by the funded by the Ocean Protection Council and supported by the State Water Resources Control Board here in California. Our approach was to field test 4 methods, and we wanted to make sure that we looked at methods that were already being used. And it just so happened there were some methods that were being used on a relatively large scale here in California. And I'll talk more specifically about the methods in just a minute. We also wanted to make sure that we weren't doing all of this in a bubble. We wanted to make sure that we brought in some experts in a number of different areas related to trash. So we developed a technical advisory committee to guide us and give us feedback. We also involved stakeholders. So one of the things that I will show you will be that we included nonprofits and other types of stakeholders in some of our testing as well. So what were the products for this project? Well, we had basically 3 products, and that is a field testing report, which basically summarizes the results we received when we tested each method. It also summarizes the results of method comparisons. Because the methods were so different, we wanted to see how well they performed against one another. And I'll give you an example of that in just a little bit. We came up with a playbook for trash monitoring. This includes the standard operating procedures for each method, and it gives a whole bunch of other information on the methods and the data collection. We also wanted to do some outreach and training, and this is part of the outreach. And again, I'm very grateful. Thank you, Suzanne and team, for asking me to speak today. So let me describe the methods very quickly. We looked at a qualitative or a rapid trash assessment. I often call this the visual method, where you walk an area of a stream or river and you give it a score based on what you see. We also did 2 quantitative methods. These are methods that were already developed and already being used. by the Bay Area Stormwater Management Association. They use a volume method where they estimate the volume of trash in a given region of a stream or river. And the Southern California Stormwater Monitoring Coalition, they actually look at trash a little bit differently, or much differently than the Bay Area group. They do tallies. So they actually tally the types of trash that they're finding within a specific area in the river or stream. We also wanted to look at developing a novel method. And in this case, we used we use drones. So, and in capturing the imagery, we also worked on developing some machine learning to identify the trash within the imagery. So the qualitative visual assessment was basically, again, a rapid assessment that you visualize the area, you give it a score from 1 to 12 based on how much trash is in that area. And it's probably the quickest method out of all of these methods to do. The quantitative method, the volume method, it's one that you walk along, you pick up all the trash and you estimate the volume of it. And it's a good method for use if you want to know how much trash is in an area, as opposed to doing something like mass, because oftentimes when you're in a riverine area, much of the trash is wet. So we wanted to take some of the variation out they wanted to take variation out by doing volume measurements. The SMC tally method, we've got, they use a pretty extensive list of items, and they basically walk along and tally all the items. They are also items that are placed into larger categories, such as plastic, glass, metal, wood, those types of things. But they also go into a lot of detail. They look at whether or not it's a cigarette, a small plastic piece, a hard plastic piece, a soft plastic piece, all kinds of different information that we collect when using this type of method. And then again, the drone method was basically used as that new method to develop and look at, and really what we wanted to do was expand the amount of area that was covered and see if we could get some information from that. And it was a really neat method, and I'll give you some of the results we got from that as well. So as many of you know, the higher you fly, the less resolution you have, the lower you fly, the more resolution you have, and battery life is a big deal as well. But you can cover a lot more area than you can do with boots on the ground. And if you can identify the trash, it becomes a really great method to use. So there was a lot of things that we did in doing this. We leveraged a lot of the tools that are currently out there, and we can go back to the same areas over and over again and assess those areas as well. So let me give you a little taste of what the methods look like as far as evaluations go. Again, we wanted to make sure that we looked at some different aspects of all the methods, and not just comparing them to one another, but also looking at how well each one of them performed individually. So we looked at things like accuracy, precision, resources, and we wanted to categorize them based on this type of matrix. And I'll show you this completed matrix in just a little bit. One of the things we wanted to do was something called an extinction curve, where basically you have a team, and I was real sneaky, I didn't tell them what they were doing at first. You have a team walk the area and identify all the trash, pick it up. Then you have them walk it again and identify it and pick it up. And you have them keep doing it until they report to you that they've got zero trash. They're finding zero trash. And what you can see here is that in a lot of cases, after the first round or into the second round, they have picked up the majority of the trash. This red line, this one that goes up to about almost 500, that actually was this site on the right that's pictured on the right. And you can see that the total amount of trash, it didn't fill the buckets. Even though there were almost 500, there were more than 500 items, if you total up through all the passes, they barely even made a dent in the bucket. So what we got out of this was that size really does matter when you're looking at trash. And if you're in an area that has some smaller trash, you want to make sure you do more passes in order to pick everything up. Some of the other sites, I don't have a picture here, but they had larger pieces of trash, and they were easier to identify and pick up in the first and second round versus having to do more to get everything. One of the other things that we did, and this was a great exercise, we had 3 different teams do the same site, and you can see the difference in the numbers, and the numbers, the variation in the numbers get larger with items that you have larger amounts of. In this case, in plastic, we had a pretty large amount of plastic items, and you can see the disparity. We also took the items back to the lab, and we had a higher number, and that we think is due partially because as we took the stuff back to the lab, there was some breakage of the items in the transport. So this was a great exercise and gave us a lot of insight into what we were doing. In the field testing report, we also have some information on trained versus untrained groups as well. Method comparisons, let me check the time real quick because I know I'm coming up really close to the end, and I've still got a number of slides. We found that volume related to that visual assessment, the qualitative assessment, the relationship was pretty high. So here's some images from the drone work that we did. And what happened was we had people go in and basically train the imagery. They had to mark the imagery up where they saw trash. They were able to zoom in and out of the imagery and identify trash within that. And you can see here, if we zoom in a little bit, you can see some other items and specifically what those items were. One of the things that we wanted to do was look at do a comparison between the drone imagery versus the tally method. And you can see here, if we do nothing, the difference in the numbers is pretty large. But if we take some of the smaller pieces out of the numbers within the tally method, the numbers start getting closer. And that just goes to show that with the drone imagery, you're able to identify some of the larger items, but you don't necessarily have the ability to identify some of the smaller stuff. We built a lot of relationships during this project. We worked with the tobacco product waste folks, and we developed a method, a tiered method for the Department of Public Health Tobacco Control Program. They're using it. They actually have been talking to us more about doing some more work and helping them develop an application to collect some of the data from this method we developed. We worked with a grad student from a university in Chicago where they were doing stream surveys, and we had them test out our methods as well to see if they were applicable in other, not just in Southern versus Northern California, but also in other states. We also developed some relationships with some supercomputing firms that allowed us to do a lot of the machine learning at a very quick rate. We worked with the United States Environmental Protection Agency. They have a method. They asked us to test their method alongside these other 4 methods at a subset of our sites, and they gave us a contract to do that, and we did that as well. So what's in the playbook? The playbook basically has everything you would want to know about all the different methods, the standard operating procedures, and something that not a lot of people often think of it, but safety considerations when you're going out into the field. There have been a number of things that have happened that we've learned a lot from in doing this project and doing this type of sampling. So there's safety information. There's also information on how to record the data and manage the data as well. So to make sure that it's standardized. And one of the things I want to end with is looking at the methods in a different way. What we did was we actually put them into tiers, and it goes from the easiest to the most complex. So tier 1 is basically that visual assessment that's saying, this area looks very trashy versus it doesn't look very trashy, to the UAS method where we're identifying plastic versus not plastic, And then the volume method, which actually puts things into larger categories. So we get volumes based on how much plastic items there were, what that volume of the plastic was versus metal and other types of trash. All the way to the tally method that goes into very specific categories and items. What you can see here is there's a variety of questions that each tier answers, and they go from general again to more specific. If you go down to tier 4, you can actually ask questions about or answer questions about, you know, is a bag ban working? Is a expanded polystyrene ban working? Or is it the amounts, are they increasing or decreasing? So here's that that matrix that I talked about. You can see we've got some different information here on accuracy and precision and the amount of resources that are required to do each one of those methods. As you're asking your monitoring question, if, for instance, you don't have enough resources to do a tally method, perhaps you need to revisit your monitoring question and ask it in such a way that maybe you would choose a different method to answer the question. So overall, findings and conclusions, again, size matters for all methods. As far as trash goes, cost is definitely varies by what method you're doing. You can use some methods to predict relationships, and you can't use them all. I only showed you one graph there, and we talk about other relationships between the other methods as well in the field testing manual. We want to encourage people to use trash density as a metric so that we can actually have more comparability across the different types of methods and across different types of regions. And the one thing we learned is training is really critical for any type of repeatability using the methods. So which method is best? Well, what we tell people is there is not a best method. It depends on your monitoring questions, your resources, and how you feel about accuracy, precision, and all those other parameters that are involved with doing trash assessments. So that's it. If you have any questions, please feel free to ask them in the breakout session, as well as visit our trashmonitoring.org website. The reports are available through that website, and you can also reach out to my project partner, Tony Hale, or myself at SFEI. So thank you very much, and I'll turn it back over to you, Suzanne.
Thank you so much, Shelly. That was fantastic. It's really impressive that you've been doing this since 2007. So I have lots of questions jotted down, but for now, let's go ahead and move on to Wynn's presentation. So Wynn, you should be able to share your screen. Go ahead. Awesome. Glad that worked.
All right. Thank you so much. I just dropped a link to my presentation in the chat in case anybody likes to view it that way. I'm gonna be switching in and out of different tabs to demo some projects that I'm working on for everybody. So I'll just leave it at this view. This is a photo of me doing what I love, diving into trash and finding buried treasure. And that's what I do with data too. And my real obsession in data comes when I find those treasure data sets that are out there. And there are many that I'll be talking about, many ways to look at them. I don't do anything alone. I have many collaborators, and I couldn't do any of these projects without them. Pretty much any paper I write is like 10 co-authors or something like that, you know. So I just, and many acknowledgements, many people here. I just really appreciate all the folks who have made these projects possible, and my funding. And really where a lot of this inspiration happens is at trash datathons. And you're gonna have the opportunity to attend a microplastics datathon as a breakout group as part of this. And so I'll discuss a little bit about what that is. When I think about data, I think there are at least 3 basic steps for data management and utilizing data. First, you need to get some data. And then you want to visualize the data to see what you got. And then you want to interpret the data. And there are several projects that I'm going to be talking about that are steps along this data processing pipeline that I've been working on. So the first one is the trash data treasure trove. This is actually my oldest project, and it is it is a hoarder's nest of data, as I like to call it. I'm a trash data hoarder, and when I see a data set that I like, I snag it up and I save it and I put it on here, and I try to develop metadata associated with it so that it can be more useful and user-friendly. And a big topic at datathons recently has been how are we going to quickly navigate this data? Because it's a lot. I mean, there's hundreds of data sets in here, and we want to quickly be able to find the data that we need. Because sometimes you don't even need to collect new data for the questions that we're managers, policymakers, scientists are asking. There might already be a data set out there for it. And so it's best for us to be able to build on works that have already been done. And one project that has already been done with this, an undergraduate did their undergraduate thesis on some data sets from this trash data treasure trove. They really dug through the hoarder's nest and found some treasure in there. And how you can be involved. Actually, if you want to be a collaborator on this project, I would give you access to uploading data on here, create your own component in here. You can share data, help us curate metadata. Use this data in your own projects. The next one is clean up our world. So we're moving on to data visualization now. And this project, so we have so much data, it's actually a challenge to visualize it. One of the main ways that we want to visualize data is on a map so that we can find out where it is. And we have worked with some computer scientists in our department to create a simple software for visualizing trash data that's been published, and you can get the link to those publications. And this software, you can see a instance of this concept with a bunch of points on a map, and they're visualizing where the trash is around the world. And you can use this code. It's open source, so you can, if you need to visualize a big data set on trash, go ahead and grab the code and use it for whatever you'd like. Want people to get up and running as fast as possible with trash data. That's a big goal of mine. The next thing is the trash taxonomy. So now we're getting into data interpretation. All right, I have a bunch of points on a map. Okay, how do I understand what do they mean? Is it a problem? Isn't it a problem? Is it all water bottles? Is it Coca-Cola bottles? And one of the things that's really challenging in data analysis when we have all of these data sets, all of these organizations collecting data, is that and with no current standard terminology, we then have to figure out a way to merge all of these different data sets to one another. So for example, you might have one group collecting data on the number of utensils that they see in the environment. The other group is collecting data on the number of forks, knives, and spoons that they see in the environment. Obviously, those two data sets are related to one another, right? The number of forks, knives, and spoons, if you add those all up, that equals the number of utensils. But you have to develop a relational database to actually be able to do this efficiently without having to reinvent the wheel every single time you want to bring a new data set in and merge it to a different one. And so that's what we've done with the trash taxonomy. This is part of the Water Quality Monitoring Council data management team. We had several collaborators on this, and I can just real quickly, like I said, I was going to kind of open up some different tabs. Go to the trash taxonomy website. Here you can browse the data sets that are these relational tables. These can be used. This is like fundamental building blocks for data management, for trash data management. So like the use cases are really infinite here. Honestly, I have a hard time articulating that to people sometimes because there is so many things you can do with it. And one of the things that we've started doing is we just created this like query tool where you can upload a data set and it will tell you what are the more specific items, less specific materials that you could have used instead of that word. And so this is kind of like a step, if you wanted to do like develop a new survey protocol, for example, you merge it to this data set, figure out what all of the other trash surveys, so there's 69 trash surveys that are already merged in this database. You're going to be automatically comparing your data set to 69 other data sets just by uploading it in here and being able to interpret, you know, how, like if you choose the word plastic, for example, what are you missing out on? Or if you go a little more specific, how would that merge up? All right, so we'll go back here. And this is already being used in the marine debris tracker. They have this is their exact challenge right now. They have all these survey lists in there. I think there's like over 20 different trash survey lists in the marine debris tracker app. And they need a backend that can actually merge all these data sets to make a unified data set. And so they're collaborating on this project. You can grab these reference tables, use them, develop new query tools. There's a lot that can be done. We're working on some really interesting projects with these data sets now. The next one is the reporting guidelines. So this is microplastic specific. The other projects that I've mentioned previously are macroplastic, trash, and microplastics. But this is microplastic specific. We got together a group of researchers from around the world to discuss what we think the critical criteria are whenever you're publishing a study on microplastic that you need to report about your methods so that that study can be reproducible and comparable to other studies. And this really came, the inspiration for this one came from myself and several other co-authors who had been reviewing papers on microplastics and just not able to make any sense out of the studies. Like, what did this person do? How does that compare to what this person did? And so we essentially developed these criteria based off of all of those experiences that we had, and we voted on them. And this is currently being used in many other studies. It's being used. I know like ES&T reached out to me, said thank you for this. Their editors are using this as criteria for publications right now. I've seen it on several grants as requirements for reporting. So it's being used quite a bit. I'm really excited about it. There's a cool, I love making websites because I feel like that makes everything simpler. Just go to this URL address and you'll see what's up. Anyways, there's this nice like mind map that you can use. So if you, to navigate the data set, so it says like microplastic reporting guidelines, microplastic quantification, and then you can like, boom, bring it out and see all of these different pieces of information that tells you a little bit more details about what you need to report. Or you can simplify it. Oh, I don't care about microplastic quantification. I'm not doing that. I want to look at identification or whatever. So this kind of helps to like navigate this very large list of requirements that are there, and a cool part of the project. Open Speccy, we just published the Open Speccy manuscript. Really excited about that. It is a software for analyzing Raman and FTIR spectra. And what this does is we've really focused in on microplastic spectra, and we want people to be able to analyze microplastic spectra as quickly as possible for free, and get an accurate result. And so basically, like I just uploaded the file, it does automatic processing and then tells me what it is in the end. Okay, that's LDPE or HDP, some type of polyethylene. And we've done a lot of validation on this tool to make sure that it's like above 90% accuracy. If you upload any spectrum that matches something in here, it's going to give you the right result over 90% of the time without tuning anything. That's really our goal, is kind of like an automated pipeline with OpenSpec. And jumping off of that, I'm going to be starting with the More Plastic Research Institute, and I want to collaborate with you. I'm kind of diving in on the spectroscopy side of things and like image analysis. And so we have a mapping FTIR, which I'm really excited about, fluorescence microscope, and we have a clean lab and lots of really professional researchers in our lab that can work with you. And so anyways, if you need any microplastic lab equipment or want to collaborate, want to, apply for grants together, all that kind of stuff, like that's my gig for moving forward. And that'll be starting in September because I'm graduating. I can't do anything right now because I'm writing my dissertation, but Shelly Moore, you could email her if you want to get into the lab now, and she will field, she'll help you get into the lab and get up and running. All right, so in closing, everything that I've shown here is open source. That means the code for all of these tools you can have. I will give it to you. And I don't want anything in return besides for you to use it. Also, this presentation is open source. I shared the link. And we're gonna have a microplastics datathon in one of the breakout rooms, and we're gonna talk about how we can improve open source practices in microplastics research. And we're gonna discuss what tools we need. So it's gonna be kind of like a big picture brainstorming session. I'm really excited because there's so many great minds in the room, and I just want to know what's next. As you can tell, I get really excited about new projects. So who knows? Maybe something that you propose in here will become the next thing I spend a year of my life on. Or maybe we'll form some new collaborations as part of it. So that'd be cool too. Anyway, I'm really active on Twitter, Win_open_data, OpenSpeccy, Great Lab UCR. You can email me. So thanks.
Great. Thank you so much, Win, and also Shelly, for great presentations. It's so exciting to hear about everything you're working on, and also exciting to hear that you'll be working from the same place soon, too. So that's fantastic. I have to say, personally, we are thankful for OpenSpeccy pretty much on a daily basis. We've had an FTIR for a couple of years now, so thank you for that. So now we're going to go ahead and get started with breakout rooms. I am setting up 2 breakout rooms, one which is assigned to Win and the other which is assigned to Shelly, to lead discussions on the work they presented, or if you have ideas that you want to share with them, as Win suggested. We're going to keep it pretty casual and open as far as discussions go. And we have quite a bit of time for discussions. We can let it go for about a half an hour or so, and then take a bit of a break before we gather again to hear from Western Washington on the modeling work that they've been doing. So I've set it up so you should be able to choose your own breakout room. If that doesn't work for you, send me a message in the chat and let me know, because sometimes funny things happen. But I'm going to go ahead and open the rooms. Back, everyone. With the last 20 minutes or so, we're going to hear an update on Bayesian modeling approaches being used at Western Washington University to look at the risk of tire wear particles in combination with other stressors to aquatic ecosystems. And so I'm going to go ahead and hand things over to Emma Sharp so she can tell us all about it. Emma, are you there?
I am here. Can you hear me?
Awesome. Yep, I can.
Okay.
I'll go ahead and let you share your screen.
Yeah. Okay. So today I'm going to be talking about our microplastic risk assessment for the San Francisco Bay. My name is Emma, and my co-authors on this are Skylar Elmstrom, Erica Whitney, and Dr. Wayne Landis. Now, some of this is going to sound familiar to many of you, but I want you to hang on till the end because there's some new exciting things to look at. I also want to mention that this work is supported by a National Science Foundation Growing Convergent Research Grant, with additional data support from the San Francisco Estuary Institute and Oregon State University. Okay, so today we're going to be talking about the reasons and goals for this case study. We're going to be looking at the data set we're using for the microplastic field data. We're also going to be going over the approach we're using, and we're also going to get a chance to look at the results, just initial results. And then I'm going to be talking about next steps. So why do we need a risk assessment? Well, as we know, public awareness and concern around microplastics is absolutely growing right now. And this is partially because the data on microplastics is growing. And currently, there are no state-of-the-art risk assessments for microplastics that have been published. The goals for this case study are to complete ecological risk assessment for microplastics in the San Francisco Bay that can be used to inform stakeholders of the risk that microplastics present and to develop a framework that can be used in other regions for other case studies. So the data set we are using is from Sutton et al. 2019, and it was done by the San Francisco Estuary Institute, which is based in San Francisco, California. And they monitored microplastics in 5 sample matrix: stormwater, wastewater, effluent, sediment, surface water, and prey fish. And then they also characterize microparticles by composition, size, shape, and color. So this is a really useful data set. I think it's an excellent thing that we are getting a chance to use it, and we're really excited to work with the San Francisco Estuary Institute. The method we're going to be using to conduct this risk assessment is the Bayesian network relative risk model. This model, these methods have been published from many different sites and a variety of different types of risk assessments, including adaptive management, human health, meta population dynamics, and it's all summarized in Landis 2020. And I encourage you to check that out if you want to learn a little bit more about this method. I'm also going to be breaking down how we use this method in the next few slides. So of course, the first part of this is to actually pick a study site. And we picked the San Francisco Bay primarily because of the microplastic field data set that we had there from the San Francisco Estuary Institute. And the San Francisco Bay is a really interesting area. Lots of, I think 40% of the fresh water flows through there. There's 7.5 million people who live in our study site, so a lot going on. And the next part of the Bayesian network relative risk model is to actually break our study site into smaller regions called risk regions. And you can see in this map here, we've broken down our study site into 4 risk regions based on hydrological unit level 8 watershed boundaries. So we really wanted to consider watersheds and how that might affect microplastics in those watersheds. The next part of the Bayesian network relative risk model is to develop a conceptual model. And you can see at the top of this slide that the framework we use to develop our conceptual model considers the relationship between sources, the stressors, the habitats, the effects, and then the impacts to the endpoints. I'm not going to go into a lot of detail, but I want to point out a couple things. We broke our stressors down into microplastics, nanoplastics, and tire wear particles. We did this because of differences in sources, toxicity, and fate and transport. You can also see our 5 endpoints under impacts. And I also want to point out that it's important for us to consider particle characteristics, sorb contaminants, and water quality parameters and how those might directly affect our stressors as well as affect our endpoints and their ability to deal with the exposure to stressors. So from our conceptual model, we take our conceptual model and use the data that we have to create Bayesian networks. So in this situation, we have decided to split our conceptual model into 2 Bayesian networks, one for microplastics and one for potential tire wear particles. In this situation, we have categorized microplastics as plastics that have not been categorized as rubber or potentially rubber. So the San Francisco Estuary Institute characterized their microparticles using Raman or FTIR spectroscopy, and they categorized plastics either in their various compositions, but included categories called rubber and potentially rubber. And those are the ones we are using to indicate potential tire wear particles. So today, we are going to just look at our potential tire wear particle Bayesian network. And this is where this is some exciting, very initial and preliminary results to show. It's kind of exciting. We were able to actually put some data into our Bayesian network. Now, these are very initial kind of practice attempts with this model. So it's very exciting, but it doesn't necessarily show the results we will present at some future point. What I want to point out is, in the middle here, you can actually see the data that was collected by the San Francisco Estuary Institute for stormwater, sediment, effluent, and surface water. And then in addition to collecting that data, the San Francisco Estuary Institute also indicated the wastewater treatment plant system that was used, so either secondary or tertiary, when they were collecting the effluent from wastewater treatment plants. In addition, they also looked at season when they were collecting surface water to look at the microplastic particles in the surface water. In California, there are only the dry and wet season. You can also see that I've broken this data into our 4 risk regions. So San Francisco Bay, which is the Central Bay, San Pablo Bay, Coyote Creek, and Sassoon Bay. And then we also have some really exciting data from Oregon State University. This is the Daphnia mortality toxicity data that was done by the Harper Lab. So this is really exciting that we get to include this in here too. And this is really what makes this all kind of work, which is really exciting. So I wanted to show how this works a little bit. I can also, so I have inputted the data, but I can also choose and select what states I want our boxes or nodes to be in. So in this situation, I've chosen San Francisco Bay, and you can actually see how our distribution changes in each of our states. So I'm going to click back to the previous slide, and I want you to look at, maybe pick one box to look at, so sediment or stormwater, and just see how it changes. So you can actually see the distributions changing because I've selected one of our risk regions. So it's really interesting. We can actually tell some of the things going on just in the San Francisco Bay. So now I'm going to select secondary treatment for wastewater treatment plants and see how that changes. So when I do that, you can actually see a couple things. I'm going to go back here for a second. For the San Francisco Bay, you can actually see that most of the samples that were collected for effluent were from secondary treatment systems. So this tells us a little bit about our data, which is really useful. Again, you can see a similar trend. Most of the secondary treatment samples that were collected from the San Francisco Bay region. You can also see some changes in the distributions within the middle column, so within stormwater, sediment, effluent, and surface water. And then if I selected all 3 of these, what I wanted them to be, you can see again changes in some of the distributions of particles. One thing I do want to point out is, and some of you may have noticed this, the Daphne mortality node or box over here is actually not changing. I'll go back through some of the slides, but it's not changing despite the different inputs I'm putting into our model. And the reason that we're thinking this is likely caused by is basically the field concentrations are incredibly low. So I wanted to show you the actual model, so the dose response model that we put into this Bayesian network. So the way we put that model into the Bayesian network, you can see down in the left-hand corner, is the box that we basically ask the model to take in. So at the very bottom, you can see something called the equation. That's the equation for this dose response model. It doesn't look very pretty, but I assure you that is the equation. And basically it takes that, it looks at the concentrations that we've input from the field, and it determines what the response would be based on that. And so you can see that where the arrow is pointing is basically the highest field concentration we have collected in particles per milliliter. So it's very, very low, and it's actually not showing any response on this dose response model. So we're assuming, at least in this particular case, that's why we're not seeing any of the changes as we continue to put different inputs in, even we're not seeing any change in this node. Now, this may change as we continue to build on this model. So we're planning on adding more data into this model and really looking at different things. So the Oregon State University labs have a lot of concentration response data we're planning on adding in for different organisms and different responses. So it won't just be the Daphne mortality box that you saw. We're also planning on adding co-contaminant and water quality parameters into this model. And we want to add those in because then we can look at both potentially how those co-contaminants are affecting the dose response models for the microplastics, but also how they compare directly to the microplastic toxicity. Does it actually matter as much if the water quality parameters are not correct for the organisms living there as much as the exposure to microplastics? So we really want to look at that in our model. And then OSU has chosen some priority pesticides to look at and begin co-contaminant water quality studies, or sorry, tire particle toxicity testing. And then finally, we also want to consider the endpoint abundance data. So the specific data for our endpoints, our organisms, and where they are in our study site. So where they are in the different risk regions and what that exposure is going to be. And then finally, I just want to acknowledge everybody who has worked really hard to make this model where it is right now. And I really appreciate it. And yeah, does anybody have any questions?
Yeah, we do have a few minutes for questions before we close up for the day. Thank you so much, Emma, for that great presentation and all the work you've been doing. Yes, looks like you've gotten some rounds of applause. Does anyone have any questions? Scott, go ahead.
Great presentation, Emma. It's really cool to see how this project has developed. Really quick question. So when you're doing the risk characterization and comparing the concentrations of tire wear particles that have been monitored versus what you see in laboratory experiments, are you looking at the same size ranges with those methods?
Yeah, so that's actually something I just took a look at, is the distributions for both the field particles we're collecting. I think that's something we are going to have to look at a little bit closer. It's something we're going to have to look at both data sets for. But yes, it's certainly something we want to consider is, are we looking at the same size distribution or is it close? So that is something.
When it looks like you have a question as well, go ahead.
Thanks. Yeah, that was really interesting. I'm such a novice with these Bayesian networks, but I'm really curious about them. So maybe I could ask you this really nerdy question. I wonder, how do you bin continuous data? How do you decide where those bins go for the concentrations?
Yeah, so in this case, we specifically for the microplastic field data set, we use Jinx optimization in R. So I basically asked R to take the data sets, give me 4 states and break it down into the clusters that make the most sense for this data. And that's what I chose for the field data set. For something like the dose response curves, we actually consider that based on the response. So do we want 10? So first we want to break it down into 10% and then 50 and then, yeah.
Okay, cool. Yeah, thank you. I'm gonna go research some more on that.
Yeah, and then of course you do have to play with the nodes a little bit, as I'm sure you've maybe discovered with continuous data.
Yeah, that's really interesting. Thanks.
Emma, can I point out something?
Please do.
I have in the slides.
Yes.
We can actually go through and set the last node. Let's say, how much of all this do we have to get like a low response or a high response, and actually calculate backwards what the concentrations have to be to get that response. And we just haven't done that yet because we're still making sure we got it going correctly from what we have now to the response. But we can actually make the model run backwards. So
it's a very cool tool.
How high the concentrations would have to be to actually see that response. And my question is, mostly to the rest of you, do you think we're actually, my suspicion is that we're undercounting the particles. Like when someone says, I have 1 microplastic particle per grab, you know, that's really low on a per unit mass basis. And given the inputs we see of the raw material, do we actually believe that we are counting accurately the concentration of field? That's something we desperately need to know. Because right now we're so focused on what we see in the field. And I think it's like the early days of doing dioxins and stuff, is that our detection limits were so high compared to the action level. And here, I think our detection limits we're just not seeing the plastic. I think that's what's going on. And if not, we really need to know that too. So those dose response curves, we looked at that data. Of course, we're working very closely with Brian and Stacy and Suzanne, and they're doing everything that we ask them to do as far as, you know, having the right numbers and the right spacing. And those are the curves that we're getting. We really have high confidence in those curves. Yeah. Yeah.
Wayne, I think that's a really important point to bring up, and I completely agree that we are undercounting, especially as a lab that does a lot of pulling microplastics from fish guts. We're definitely missing quite a bit of it.
In the fish.
Mm-hmm.
Get into the organisms so we can see them there.
Mm-hmm.
So does that make sense? As our collections really, do we have a mass balance between what's input, what's getting in the fish? Are we describing what's in the environment? And I'm unhappy that we're not measuring it better. Yeah.
We'll get there.
Yeah, absolutely.
So since we only have a few minutes left, I'm going to go ahead and share my screen again. Just had a few announcements and kind of closing remarks as we finish up today. This is a really great meeting. Thank you so much again, everyone, for coming today. It's short but sweet. If you have any follow-up questions or thoughts, feel free to contact me or Stacy or Wayne or any of the speakers today to follow up on things. So as far as upcoming meetings, we will be having another teleconference in October. Probably on October 14th. So tentatively save that date for now, and we'll be announcing, you know, who will be presenting, what the topics will be for that. I'm sure we'll have another update on modeling approaches as well. And then in 2022, the hope is that we can be three-dimensional again and actually meet in person. Hopefully in conjunction with the Pacific Northwest CTEC meeting. So I'm not sure where that's planned to be yet. Stacy might know, but stay tuned for that. And then as far as other meetings, in September, there's going to be an MCON meeting, which is being hosted by Washington State University, and that is an international conference on emerging contaminants. I will post the link to registration in the chat for that, but there will be a lot of talks on microplastics. I believe Dr. Chelsea Rockman is giving one of the keynote talks there, so definitely worth tuning in. And then for Portland CTAC, which is coming up in November, and that's a hybrid meeting. So some of us will be in person, depending on how things go with the pandemic, but there's also an equally good opportunity to join online. There are quite a few sessions on plastics, nanoplastics, microplastics, risk assessment, modeling, whatever your favorite flavor of plastic pollution is, it will be represented there. And so I've listed the sessions that will be at Portland, and then I'm probably missing a few, but you can go to their website if you have questions about that. And let's see. So I'm going to go ahead. Oops, I didn't mean to close that out for now. Let's try one more time. There we go. And then one last announcement, and this is from Shelly. There's going to be a microplastics monitoring subcommittee kickoff webinar on September 30th. And I've got the registration for that link here and that meeting here, and I'll also post that in the chat as well. So lots of opportunities to join in on more discussions about microplastics, nanoplastics, plastic pollution in general, and hopefully we'll see you all at some of these future meetings. And let me just go ahead and make sure I'm able to post those few links in the chat for you before we close out. There we go. Does anyone have any questions, things that you wanted to ask various presenters before we close out for the day? I've also provided a link again to that hearing that's occurring. It's actually ongoing now, and so you can drop in if you're interested in the House hearing, as well as links to the various meetings that are coming up. So, well, if no one has any further questions, thank you so much again to everyone for joining us today. Thank you to Shelly and to Wynn and to Emma for presenting. It was great to see you all, and hopefully we're getting closer to being able to see each other in person at some point. So thanks everyone, and have a great rest of your week.
Thank you, Suzanne.
Thanks, Suzanne.
Bye.
Bye.